Hi, everybody. Welcome. This is my first time in the new Wilf Center. It's lovely. So thank you to the Wilf family. And hello to those of you looking online. I'm pleased to introduce Dr. Todd Druley from Washington University. He is a longtime friend of the Division of Epidemiology here in the Department of Pediatrics, as well as the Epidemiology Committee of Children's Oncology Group. He received his undergraduate degree from University of Illinois Champaign, and then did the MSTP program at University of Illinois Chicago, where he got his MD, obviously, and a PhD in molecular genetics, and then has been a resident and fellow at WashU since. He's been remarkably productive as a junior professor, has over 25 publications, is the recipient of a K Award, as well as Doris Duke, Hyundai Hope and Wheels, and then Alex's Lemonade Award. So, like I said, remarkably productive, and he has been working for the past few years with Julie Ross on her infant leukemia samples, has some very interesting findings to share that are very relevant to anybody interested in cancer predisposition. So, without further ado, Dr. Druley.
All right, well, thank you very much. Hopefully everyone can hear me all right. It's a pleasure to be here. I've got a lot of great colleagues here. Logan especially is one of those, and so it's a real pleasure to be here. It's a beautiful facility, and it's nice to have the chance to present. So I have no conflicts to disclose, and we'll get right to it. So the fundamental question that I and many other people ask is, why do children get cancer? And so historically, we can't really explain the incidence of pediatric cancer solely through cancer-causing mutations, P53, APC. There are some mutations. Retinoblastoma, they'll get cancer early in life. A lot of that is the contribution of ionizing radiation on the retina, Wilms tumor. But by and large, you can't explain the incidence solely through highly penetrant cancer-causing genes. There's been tremendous amount of research looking at somatic mutations or environmental exposures. And it seems like right now the conventional wisdom says that we have the two-hit hypothesis that tends to align more with the highly penetrant genes. For less penetrant genes, it might take up to 6 or 8 different mutations for a normal cell to transform to a malignant cell. And then as the next generation sequencing era has evolved, and we've historically focused on chromosomal rearrangements in the affected children themselves, but we are now finding that these chromosomal rearrangements are often found in healthy people that don't get the cancer. So it's not to suggest that these things aren't involved, it's just to say that they're not the whole story. So for the purpose of this talk, I just want to make sure we have clear definitions as I talk about things through. So mutation, when I talk about mutation, it's a change to your DNA sequence after birth. So typically this is going to be only found in your cancerous tumor. It's a somatic event. But when we talk about variant or variation, this is a change in the individual's DNA sequence from the human reference. And so if this is a constitutional change, this will be in every cell of their body. So as we talk about variation, I'm primarily going to talk about germline and constitutional sequence changes. So this was from a review a couple years ago in Science, and meant to simply show you on the y-axis here we have the number of somatic mutations, and the x-axis we have a list of different types of malignancies. And so the real takeaway point here is just to again show you that quantitatively, qualitatively, there are differences in the amount of somatic mutation between pediatric and adult cancers. And so and again, can't really explain the incidence of cancer solely through exposure histories. So in the lack of somatic events explaining these cancers, we have thought about germline events that might contribute to this. Another interesting piece of data from the SEER group is showing that over the last 40 years, there's been a very steady increase in the incidence of pediatric cancer. Not clear what's driving that. Is it better detection? I would argue against that. I would think if it was better detection, you'd see spikes where a certain technology came online, CT scanner, MRI, something like that. It doesn't really bear out in that data. So potentially it's due to additional exposures. One thing I would just throw out as pure speculation is that when we look at ethnic-specific data, you see pretty much the same trends. And so if we're talking about germline events that are driving malignancy, it's not going to be a single gene or a single mutation, typically. We would have found those by now. But if this is a constellation of multiple genetic variants, then if we talk about admixture, right, this is the blending of different genetic backgrounds, then the last 100 years, the Earth has the greatest amount of genetic admixture that it's ever known. So populations that were historically segregated are now able to freely combine, and we're getting new blending of very historically isolated genetic backgrounds. Again, pure speculation, it's interesting to think about. So again, the hypothesis will be, are children getting cancer partly because of inherited profiles of rare or damaging genetic variation? And, is there any data to support this hypothesis that exists? I think there is. There's various things you can point to. One was an interesting study on acute lymphoblastic leukemia from the early 2000s out of Scandinavia. So because they have such good lineage tracing in Scandinavia, they were reporting that the odds ratio, if you're not a twin, but you have a full sibling that has ALL, your odds ratio is actually 7 to get ALL. And if you are twins, it's an incredible odds ratio of 137, suggesting that there are other events that you've inherited that tend to increase your risk of malignancy. This is a paper I found particularly interesting from a few years ago out of the University of Cincinnati. So they followed 300 of their pediatric cancer survivors and asked them a battery of questions and found that 29% of them met criteria that they would consider to be consistent with a pediatric cancer predisposition syndrome. And when they considered the children that had passed away and thinking that most pediatric cancer predisposition syndromes confer an increase or a worse outcome, then they said as many as a third of the children that they see would have a cancer predisposition compared to only 5% for their adult cancer survivors. And there's a growing body of literature suggesting that birth defects confer a higher risk of childhood cancers. Just flipping through some of the different papers that are out there, there's probably more by now. But there's a variety of studies, nice epidemiology studies looking at this. And so the punchline for this is that the relative risk, depending on how you define a birth defect and what birth defect you're looking at and what type of cancer you're looking at, it seems as if the relative risk is anywhere from 1.5 to maybe 6 for an increased risk of early childhood cancer with these birth defects. Again, suggesting that there's something in the germline that's increasing the risk of cancer. So I want to talk a little bit about rare variation, because like I said, if this was a single gene or a single mutation, we probably have found most of those by now. So we're probably talking about multiple rare events that align within the same individual. So what do we know about rare variation in general? Well, the definition currently, it's somewhat different at different places, but by and large, people will accept less than 1% allele frequency in the general population. So we know that the rare variants have greater effects, an average odds ratio of 3.7. These disruptive rare variants are more likely to act in a dominant fashion. And so, I'm not a statistician, but the statisticians always say if you're going to find one genetic event that's driving a particular phenotype, you have to have 5, 10, 15,000 people. Well, that's typically more than the number of pediatric cancer cases on the earth in any given year, so we won't get very far. And one way you can overcome this, the rarity of these events, is to say, I'm not interested in any one particular hit. I want to see where do the hits pile up in the people that are affected. So it doesn't have to be the same genetic change. It just has to be a genetic change that causes dysfunction in a critical gene or a critical pathway. And this is called a strategy for collapsing rare variants into sort of a larger genetic locus. So just stepping back a few slides to sort of make sure we all understand how rare variation thinking has come around. So it started from the Human Genome Project, which was finished in 2003. So we all know that we have 3 billion nucleotides in our haploid genome, so every cell should have 6 billion, 3 billion from dad and 3 billion from mom. And the major project that stemmed from this was called the Haplotype Map. And so this was meant to document the amount of genetic variability in the genome from 270 individuals at very disparate global locations. Now, mathematically, that said we're going to detect roughly 99% of all the genetic variation that's present at 5% or greater in the human population. So at the time, that was the limit of definition for rare, was 5%, and that was used for quite some time. So during the Haplotype Map project, investigators kept seeing these same SNPs. Remember, they're common, so a significant percentage of their study participants are going to have these SNPs. And the idea started to evolve that there's probably a relatively discrete amount of functional variation in the human genome. And so your phenotype is less dependent on your genetic profile and more dependent on your environment. And this was formally termed as the common disease, common variant hypothesis. So you could have the exact same constellation of genetic variants, but if you lived in Scandinavia and you were a non-smoker and you did a lot of cross-country skiing, you would have a different phenotype than somebody who lived in Zimbabwe and ate a completely different diet. And so that was the idea about this. But if we think about this, and you have a genetic variant that gives you a bad outcome, then how can it become common, right? By definition, the people that have these damaging genetic changes should have less fitness. So they have fewer children, they live shorter lives, and these are over generational time spans. So even if it started out common and you said, you know, the genetic risk factors for obesity were irrelevant 1,000 years ago because it didn't have the opportunity for that, and so now they're actually causing some selection. But even if that happens, they should become rare. So that's why rare variants typically have a greater effect size. And so it seems a little implausible to solely pin these complex phenotypes on the environment. And so, but this led to the genome-wide array era, and genome-wide arrays have given us a wealth of really interesting observations, and I tend to think of the genome-wide array as a street lamp shining a cone of light on a big genetic region that's imparting some percentage of phenotypic variance for whatever trait you're looking for. But typically, these regions of the genome are anywhere from 1 to maybe 10 megabases. And there's a lot of genetic information in most of those regions, and it's hard to distill that and say, this is the linchpin to this complex phenotype. And I think that was demonstrated nicely a few years ago in Nature Genetics. There was 3 labs using the latest genome-wide array technology at the time to study the genetic influence of adult height, which ranges 25 to 30 centimeters is sort of the range of adult height. So collectively, the 3 labs had studied over 600,000 genetic loci and 55,000 individuals, and all of this data, one of the biggest studies that you could imagine, identified 55 genes that did not correlate at all with any of the prior genes identified through linkage analyses, and cumulatively could only explain 1 centimeter, about 4% of the difference in adult height. So the authors of these studies agreed that potentially rare variation was more important for adult height, and there's probably other factors outside of the genome itself that influence height. So now we're in the next generation sequencing era, and we've seen a precipitous decline in the cost of sequencing a genome. So in 2007, when these sequencers were introduced, the cost of sequencing a genome broke Moore's law, and we're down now to where we can do a whole genome. You know, depending on where you're at, you can do it for $1,000 to $5,000 in just a few days. But the bottleneck, and some of the discussions that we've had today, is that bioinformatics has not had the same breaking of Moore's law. So nobody really cared that doing the bioinformatic analysis of the first genome cost $75,000 to $100,000 because the genome cost millions to $1 billion. But now if it costs you roughly the same to do the bioinformatic analysis as it does to do the sequencing, it feels outrageous. And so that's where the big bottleneck is in these computational structures. So you probably maybe some of you have seen this type of a graph before. And so what we have here on the x-axis, we have the frequency of any genetic change. And on the y-axis, you have the relative effect of that genetic change. And so I'll use the cursor here. I think I can. Yeah, here, am I there? Here I am. Okay, so we have the relative frequency of any genetic change, any single genetic change here on the x-axis versus the effect that that single genetic change confers. So we have the genome-wide array studies. Remember, most of those are common variants. So the impact to any particular phenotype that they confer is modest to low. But now we talk about very rare genetic changes. We're into the era of, or the range of Mendelian diseases. So think of sickle cell disease or phenylketonuria or whatever Mendelian disease you want to think of. And that's not really what I'm trying to propose here either. So I'm more talking about genetic changes in this. Did I screwed it up. It's unhappy. Yeah, okay, so operating in this range. So we're talking about not high effect rare variants and not super rare variants. I mean, they might be, but they work together. So a single variant by itself probably doesn't have much of an effect, but if you aggregate multiple rare variants, you're going to increase the effect that the constellation of the variants has on the phenotype you're trying to study. Okay, so this was, I think, nicely demonstrated a few years ago by Jim Lupsky and colleagues at Baylor. They called this clan genomics. So this is figure 1 from their paper, and it shows this ancestral pedigree here. So the people at the very back, the very earliest parts of this group here, you can see there any genetic variability that the present-day people have that came from their ancestors back here has a very low effect. It's been allowed to persist in the population, so it doesn't do much. And as we get closer and closer to near time, the genetic variability that the descendants have has a greater and greater impact on their personal fitness, and particularly if they have any de novo hits that are unique to them, that's most likely to have a significant impact on their fitness. So figure 1 here, they actually say combinations of rare and inherited genetic variation will have a major impact on one's health. All right, so this led us to the rare variant hypothesis, which I tend to think about. And so these are variants less than 1%. Private mutations are within single pedigrees. And here we're predicting that complex diseases are mediated by a synergistic combination of multiple rare variants. So we want to stop thinking about those single variants and start thinking about collapsing across the whole pathway. So you can think about these models in this way. There's 2 models, and I really persist on this because I think people haven't thought about this collapsing idea quite as much. So in scenario 1, let's say this is sickle cell disease, all right? So if you take the global population, which primarily doesn't have sickle cell disease, then the frequency of the sickle variant is going to be exceptionally rare in the global population. So here we have a single variant that, So we have a single variant that's very rare, and it's in this particular coding region. But if we looked at the sub-Saharan African population, then all of a sudden, which the sickle variant's actually 25–30% frequency in that population, now it becomes common. So the terms rare and common are relative to the population that you're looking at. That's not the model that we're talking about. We're talking about scenario 2, where if you have a particular variant in a given gene, it could be rare on the global population scale, but if you look at the people that have the particular disease, you could hit that gene in any number of ways, so that the initial variant is not necessarily more common, but you have more functional variants in that gene. So this was nicely demonstrated with cystic fibrosis. So originally, people thought that delta F508 mutation was the one and only hit that caused cystic fibrosis. So if you came into the hospital and you had symptoms consistent with cystic fibrosis, you got sequencing of the CFTR gene. And so, of course, what happens when you start doing that is you start to say, this person doesn't have delta F508, even though they have cystic fibrosis-like symptoms. And now we have a catalog out of the Sick Kids Hospital in Toronto. This is a few years out of date, probably. It's over 1,000 different hits in CFTR that all confer some severity of cystic fibrosis-like symptoms. So it's a nice demonstration of that philosophy. And of course, now with next generation sequencing, more and more complex phenotypes have been shown to have an aggregate of rare variants. This is obesity, schizophrenia, different cancers, high cholesterol, I mean, you name it. So rare variants are involved in a lot of complex phenotypes. So let's get back to pediatric cancer, right? So if we have so we wanted to study infant leukemia as a particular model, looking for germline variation that was at least contributing to the incidence of infant leukemia. And why we wanted to study this was, first of all, we had a very generous offer from Julie Ross to do this work, and she had a perfect cohort to do the work, so I'm indebted to her quite a bit. But infant leukemia makes a perfect model because this is a bad disease. This is the worst of all pediatric leukemias. Over 50% of the children will die. Survivors are often left with lifelong complications in development, learning, cognition. One of the hallmarks of infant leukemia is that 2/3 to 3/4 have a translocation in the MLL, or the mixed lineage leukemia gene. So MLL is a histone methyltransferase. So it's modifying histones, and when you modify histones, the histones are important for packaging DNA. So when you modify histones, you can supercoil DNA so that gene transcription machinery can't access the genes, or you can uncoil DNA, and now the gene sequence is accessible to the transcriptional machinery. So it's very important for controlling gene transcription, particularly in the mesoderm. It's a very promiscuous gene. It's got over 100 fusion partners that have been described to date. And so the interesting thing is you would think, well, this is the linchpin to infant leukemia, but if you tried to introduce an MLL rearrangement in a model system and you expressed it at physiologic levels, you do not get leukemic transformation. You can get leukemic transformation with a sole MLL rearrangement if you overexpress it and you wait. So in a mouse model where you only use an MLL rearrangement, you can get leukemic transformation in 8 to 12 weeks typically, but an 8-week-old mouse is like a 10-year-old child. So you haven't necessarily copied the infant leukemia phenotype by doing that. And again, Julie and others have done a lot of research, and you can't explain the incidence of infant leukemia solely through perinatal or prenatal exposures. So at WashU, in collaboration with St. Jude's, they put this abstract out at the AACR meeting a few years ago. So they had done whole genome sequencing analysis of MLL rearranged ALL cases. And in their own words, they find remarkably few somatic mutations. So the idea here is that the MLL rearrangement is not enough by itself. There must be some other somatic hit that's causing this early onset leukemic transformation. And they didn't find it in their sequencing study. So again, that makes a perfect model for us to look outside of somatic events at germline events and see if we can find anything. So like I mentioned, we were very fortunate to be able to collaborate with Julie and Logan and Amy Linneberry here with their study in the epidemiology of infant leukemia. So we sequenced germline DNA from mothers and their infants. And so to date, we've done exome sequencing on roughly 70 pairs of mothers and infants from the germline. We're not sequencing the leukemias. So it's about half and half MLL rearranged and MLL non-rearranged, and it's about half and half that get AML or ALL. And for some reason, infant leukemia is much more common in Caucasians, and so to try and keep this as clean as possible, we've only sequenced Caucasians to date. So it's still a small sample size, and we're arguing that there's rare variation here that is impacting the incidence of this disease. So how are you going to make any kind of statistical argument to this? We realize that we're going to have to use a collapsing strategy, but we want to keep that data even more refined. And so what we did was we're only interested in any rare or novel genetic variant. So that means it has to be found at less than 1% minor allele frequency in the 1,000 Genomes Project. so it's globally rare. Some of these are novel to the individual or the pedigree. Has to be non-synonymous, so has to alter the coding sequence, and then it has to be predicted damaging. So Anavar is a bioinformatics suite. It has 6 or 7 different algorithms that it uses to collectively make predictions as to whether a sequence change would cause a negative impact on the protein or have no impact on the protein. None of these algorithms are foolproof by themselves. If you use multiple ones, it tends to be pretty good, but it's not absolute. But this was how we could really skim the cream off the top of this relatively small data set. So what we found right away was that the infants with ALL or AML had more rare, non-synonymous, and potentially damaging genetic changes in their germline than a cohort of healthy children. Their mothers did too, but their mothers didn't have as much as they did. So that's just distributed across the exome. So the exome is all 20,000 genes. So how do you know if any of that has anything to do with leukemia? So we had to narrow our data set down even further, and we needed to look at genes that we knew were related to leukemia. But I didn't want to be the one to pick those genes. I think that adds too much bias into the project. So we were able to use the COSMIC database. That's the catalog of somatic mutations in cancer. That's maintained out of the Sanger Institute in the United Kingdom. So anybody in the world can add their data saying we've sequenced some cancer and we have found a somatic change in this cancer, a mutation, and we're putting it into this data repository. So we asked for all the genes that had somatic mutations in ALL and all the genes that had somatic mutations in AML. And that became our candidate gene set. So instead of looking across 20,000 genes, now we can ask, are the genes that are known to be leukemia related more likely to have our rare, non-synonymous, and damaging genetic changes? And indeed they are. So these 2 panels show you the infants that do not have an MLL rearrangement. And so what this is here we get a number. So in this, you say roughly 45 of your 126 genes have rare, non-synonymous, damaging changes in the babies that have ALL. Is that a high number? Is that a low number? Who knows, right? So what we did was do 100,000 computational iterations where we picked 126 genes from the exact same kids and asked, of the 126 genes we just picked, do we see how many of those have rare, non-synonymous, and damaging genetic changes? And when you do 100,000 computational iterations, you get these nice distributions here. And so what you see here is that computationally, by just random picking, we could never recapitulate the amount of damaging genetic variation that we found in the leukemia-related genes. It's even more striking when you looked at AML. So it does suggest that not only do they have more damaging genetic variation on average, that genetic variation is aggregated in genes that are leukemia-related. Oh, sorry, I forgot to fix this. But this is, so these are the MLL positive cases. These are flipped upside down, but you get the idea. So these are the kids that have ALL. Here's the kids that have AML. It's still wildly significant. And sorry that these are upside down. And this is an independent cohort that I'll mention later. So this is not our data. We picked an independent validation set. It's the same data that I mentioned to you from the sequencing from the St. Jude's WashU project, the Pediatric Cancer Genome Project. And so you see that the amount of variation is very similar in both ours and their MLL positive cases. So to put numbers on it, they're wildly significant. Here's your p-value for ALL and your p-value for AML babies. The mothers are also significant, not quite as significant as the children compared to the controls, and the controls are not statistically significant. So unfortunately, we didn't have fathers to test, but when we look back at what's the contribution of all this variation from the mothers, it's 47%. So you'd expect 50-50 from moms and dads. We just didn't have paternal DNA to test. It would be great if we did, but the supposition here is that this is a 50-50 mix of these damaging genetic changes from mom and from dad. We cannot exclude that there's a de novo mutation, and so we tend to call it maternal and non-maternal variation, but that's only to be inclusive of potential de novo mutation. So, again, this is a lot of hits across a lot of genes, and how are you going to make any functional sense of any of this? So what we wanted to look, if we were making the argument that this is a blending of damaging genetic changes from mom and from dad, then there should be some genes where each allele, each copy of the gene has one of these rare non-synonymous damaging hits. One from mom, and we can track that one because we have the maternal sequencing, and then one that's not from mom, so it's either a de novo hit or dad passed it on. So here we looked across all the genes, not just the leukemia-related genes, and we found, quite strikingly, there was one gene that has been a compound heterozygote in 100% of the cases that have AML, and actually now about 60% of the cases that have ALL. And that gene is MLL3. So this is a homologue to MLL1 gene, which is frequently rearranged in these cases. It's also a histone methyltransferase, and there's quite a bit of data showing that it's related to cancer, particularly leukemia, and a lot is known about it in AML. So this is where the hits piled up in the coding sequence. So this is the protein sequence of the MLL gene. It's a huge gene, 4,900 amino acids. And you can see most of the hits sort of pile up down here at the, this is actually the 3' end because it's transcribed on the negative strand. And they pile up around this poorly named regions here called ring domains. And ring stands for really interesting new gene. So it's highly subjective. But that's where all the variation, or most of the variation, was piling up in these MLL negative cases. So at this point in time, I thought, okay, germline MLL3 variation is a proxy for an MLL rearrangement in kids that don't have an MLL rearrangement. So when we do the MLL rearranged cases, we're not going to see anything that looks remotely like this. And I was completely wrong. Here's what it looks like for the MLL positive cases, and these are validated hits. And so it's even more aggregated to just downstream from the ring domains in both of these. And so, again, I said this gene's a histone methyltransferase. That histone methyltransferase activity is mediated by the set domain down here. So none of the variation, and that doesn't mean it doesn't have an affected histone methyltransferase function, but none of the variation aggregates around the histone methyltransferase domain. These rings act as E3 ubiquitin ligases. So what these do is they regulate the ubiquitination of histone 2A and 2B, another histone modification important for chromatin structure. And nobody's really explored what the ubiquitination function is for MLL3, just that the rings, that's what they tend to do in any protein that has them. So we've done 3 levels of validation because, frankly, I told Mark, who's the student that did this, I thought, this is too good to be true and you've screwed something up. So we've tried to go through and be as rigorous as possible. So we did topo-TA cloning of all the patients and Sanger sequencing. We validated over 91% of the called MLL3 variants. And what I showed you on those graphs was only validated data. Since then, we've compared to public data because our control set is small. We only have 14 or 15 pediatric controls. So we want to look at a bigger control set. So again, we turn to the exome variant server out of the University of Washington. So they've got 8,600 Caucasians. And so when you count up how much non-synonymous, and we don't even know if it's deleterious or not, but it's non-synonymous mutation in MLL3, you get a rate of 0.21 non-synonymous mutations or variants per person in these 8,600. So our pediatric control is actually a little more stringent because our rate was 0.35. If you look at the infant leukemia patients, we're seeing about 3.5 non-synonymous changes per patient. So it says that an infant leukemia patient is more than 16 times more likely to have a non-synonymous mutation in MLL3 compared to the general public, and that's a significant p-value there. And then the last layer of validation was to say, can we find the same thing in that independent cohort that I mentioned? So the Pediatric Cancer Genome Project. Now, the model for cancer genomics has always been, let's sequence non-cancer DNA and cancer DNA from the same person. Let's filter out everything in their non-cancer, their healthy genome, and because we're going to presume that it's irrelevant, and we're going to only look at the somatic hits that are in the cancer. And again, when they do that, they don't find anything that's meaningful, and they publish that. What they didn't do was look at their own germline data. And so when we did that, this is what we find from their data. So it very nicely, very closely matches. These are, again, not all the same hits that we're seeing, but they aggregate in the same position within the coding sequence. So we feel as if we validated this data in an independent cohort. And if you look at where all the somatic mutation piles up in MLL3 from the COSMIC database, you do see 2 spikes of somatic mutations at the same frequencies. Here's those ring domains down here, these little ovals. So, again, I got really fixated on why are 40% of these kids don't have MLL3 hits, and it turned out they had hits in MLL2 and MLL4. So there's a lot of overlap here in these proteins. So MLL1 and 2 turn out to be orthologs of the Drosophila trithorax gene, and 3 and 4 are orthologs of trithorax related. So they have non-redundant functions even though they're homologs. And there's already been published data suggesting a role in leukemogenesis by the protein complexes that these genes anchor. So these compass complexes will regulate gene expression during early development, and so it does a couple of things. It will do trimethylation at H3K4, so that these H3K4 trimethylation is at a promoter site. So it turns a gene on or off. It's very binary. But it'll do monomethylation H3K4 at an enhancer site. So that's more of an amplitude modulator. So you can have high expression or low expression if you've also got H3K4 trimethylation at the promoter site. And then, like I mentioned, there's ubiquitination function, which is not clear what that actually does. This is schematically what these protein complexes look like. Again, they're non-redundant. You never have 2 MLL proteins in the same complex. They are the linchpin to the complexes themselves. So 1 and 2 are roughly the same, and 3 and 4 are roughly the same. And it seems as if they have to work together to execute their developmental functions, although that's not been experimentally proven. So this is probably the most interesting observation that we've made. And so here we wanted to see how much of this germline variation, again, going away from all the leukemia genes just to the compass genes that I showed you in the prior cartoon. So if we ask how much rare, non-synonymous, and damaging germline hits do we have in the proteins for MLL1 and 2 complexes versus MLL3 and 4 complexes, we know we have a lot of MLL3 variation. So it doesn't matter if we have an MLL rearrangement or not. So here's the MLL negative kids, here's the MLL positive kids, and here's the independent St. Jude's cohort. And then here's our controls. So there's a significant difference in the amount of damaging germline variation in these protein complexes, whether you have an MLL rearrangement or not. But if you have an MLL rearrangement, you do not have germline variability in the MLL1 and 2 complexes. If you do not have an MLL rearrangement, you do have a significant enrichment of germline variability in the MLL1 and 2 complexes. So again, this suggests that there is some cooperativity between these genetic lesions regulating these protein complexes during early development, potentially at the mesoderm or definitive hematopoiesis stage. And we just picked some random pathway. We looked at apoptosis. We assumed apoptosis had no role in infant leukemia etiology. And again, we see no differences in the amount of germline variability there between our 4 cohorts. So the conclusions from this work were that infants who develop leukemia will inherit more damaging genetic change in leukemia-associated genes than their mothers or unaffected kids. This germline variation tends to aggregate in these compass complexes, and this may explain the lack of observed somatic mutation because it's mediated through germline variability. And it suggests a defect in hematopoietic development. So what we're trying to do now is we're continuing our computational biology. We're sequencing more cases. We've got some international cohorts that we're adding to this. But we want to start doing functional work. And so we're reprogramming patient fibroblasts or bladder epithelial cells to inducible pluripotent stem cells. So you can just do a clean catch urine, and you'll get some bladder epithelial cells in there from a child. You can grow those up in the lab and reprogram them to iPS cells. And the reason that's important is because we have a multigenic hypothesis here. So I can't recreate all of these genetic lesions in a single mouse or a single cell line. I want to go straight to the source, and I want to go to the kids that get infant leukemia and have their germline at my disposal. So, and again, if there's variation that's important for phenotype outside of the compass complexes, we can account for that in these models. So we've made 7 clones so far from 4 of our patients, and we've got a couple control clones. So this just shows you staining of one of these clones. This is from one of our patients here that had AML, and it just confirms pluripotency with these 4 stains here. So next what we want to do is we're going to take the stem cell, we're going to make a prediction that there are defects in hematopoietic differentiation in children that have whatever children with infant leukemia have. And so we can do hematopoietic differentiation. This is done in collaboration with one of my colleagues at WashU, Chris Sturgeon. He trained in the lab of Gordon Keller at Toronto, and he's probably one of the world's experts in hematopoietic differentiation in vitro. And he can control primitive versus definitive hematopoiesis through the regulation of Wnt signaling. And so we have this scheme. We have very nice time points where we can say we're looking at mesoderm, we're looking at hemogenic endothelium, we're looking at HSCs, hematopoietic stem cells, or we can get to terminal differentiations. And so we've been able to do this. T cells will only be made through definitive hematopoiesis. So this just confirms that we're starting here with cells from lymphocytes. This is Chris's work here, but we can go through the HSC stage and we can get to CD4, CD8 positive T cells through this scheme. And this is done by a graduate student in my lab who had no idea he would be able to do this. So it's good, we can do it. So what do we want to do? We want to study in isolation the effects of the ubiquitination domain and the histone methyltransferase domain, and we've used CRISPRs to make these changes. So what we're doing is we're going to edit out the ring domains in frame so that we have the rest of the protein, so that that protein will have an intact histone methyltransferase function. And we can do the opposite. We can edit out the set domain and leave the ubiquitination intact. And so these will look like this. So the MLL3 delta ring is about a 75 kb deletion. And so we'll still have histone methyltransferase activity. And then we can see what happens during hematopoietic differentiation if we can't do ubiquitination properly. And like I said, the opposite is true. We've done a 10 kb deletion that cleaves off the set domain. And we can see exactly what does ubiquitination target by itself. So how we want to characterize this, we have colony forming and cellular transformation assays to watch the cells differentiate and look for skewing or potentially transformation. We want to look at the chromatin state. Remember, these are histone modifiers, and we think it's regulating which genes are accessible or inaccessible for transcription, and we can do that through a technique called ATAC-seq. It's sort of the most recent iteration of DNase I hypersensitivity assays, but it's a little bit easier to do. And then we want to do quite a bit of ChIP-seq, and so we'll look for trimethylation, monomethylation, and ubiquitination of different histones. And then we want to correlate all of this epigenetic variability to gene expression by RNA-seq. So what brought some of you to this talk, I think, was to talk about cancer predisposition. And so it hit me one day that I think, we're talking about infants that get leukemia have these germline profiles, so would they have a cancer predisposition? I would argue that they do. And it struck me that, at least at Washington University, we weren't providing a comprehensive resource for all the children that we see with formal or newly recognized cancer predisposition syndromes. So we've created a pediatric cancer predisposition clinic. It's a joint venture between those of us in HemOnc and our pediatric genetics staff. And so we have 3 genetic counselors that work with us. We're now integrating the GI service. They're excited. They have about 50 patients that have either FAP or different polyposis syndromes. And there's a growing recognition that congenital CNS anomalies lead to an increased risk of CNS malignancies in childhood. So our neurology and developmental services are working with us to identify which children they care for that we might need to be screening. So we try to be as comprehensive as possible. We do offer the genetic counseling, like I mentioned. We offer a full range of therapy services. We have about 50 children that have Beckwith-Wiedemann syndrome, and as you can imagine, they have large tongues. A lot of them need speech therapy. A lot of them have hemihypertrophy. So they need orthopedic support, some occupational therapy for limb length discrepancies. Some of them are nonverbal and need highly specialized educational programs, where other ones are honor students, and we don't quite understand the differences there. And then we're using this as a research platform as well. We're banking DNA from nuclear families so we can look at the parental combinations leading to not only the syndrome itself. We know why the syndrome occurred because we've done the diagnostic tests, but we want to know, why did this child develop this cancer and the rest of the children didn't? In Beckwith-Wiedemann, it's roughly 5% of the children will develop either a Wilms tumor or a hepatoblastoma, and we want to understand why, because all the children get screened the same, and we don't have good intuition as to which ones are at the highest risk. So we want to facilitate some more genetic research into these different formal syndromes, and then we ask each family to provide to us longitudinal information. They get a 15-page questionnaire once a year so that we can back up our genetic search with demographic and phenotypic information where it's appropriate. So some of the conditions that we see, it's a long list, pretty inclusive. We have a lot of kids that have Dicer 1 syndrome for some reason, about 30 children with FAP. We've got some hereditary neuroblastoma where we found familial hits in the ALK gene, ALK, which is a new finding in neuroblastoma. So we take all comers. Just notably, we don't see children with NF or Down syndrome because they already have clinics, so don't think we're excluding them. They're being well cared for. And I would expect that there'll be a growing amount of referrals because we're doing more sequencing for more conditions. I think it's likely that somebody will get sequenced for autism or epilepsy or some other condition, and they'll find some incidental findings that might be concerning for cancer. And so where do those children go? Who can they talk to? So we'd be happy to talk to those families. I have no expectation that we're going to know what to say, but at least we can identify who they are. We can follow them longitudinally and hopefully over time add some salient information to help screen these children effectively. The brochure looks like this, and nice and glossy. And with that, I'll wrap up. And a lot of thanks particularly here to Julie, Logan, and Amy, who've been great collaborators in this. I appreciate your attention, and I'll take any questions. Bringing a microphone there.
Great talk, thank you. Have you looked for the occurrence of these MLL changes in kids with standard risk ALL or non-infant leukemia?
Yeah, I get that question a lot. We haven't yet. We haven't yet. I mean, those data sets are not easy to come by all the time.
And then your assumption was that half this variation, this increased amount of variation in these kids was maternally derived and half paternally derived, but you don't have the paternal data yet. Is there any model in which the other half didn't come from the parents? Does a maternal side somehow set these kids up for higher rates of new mutations?
Usually people want to know about the de novo mutations, and so we use the autism literature there. There was a nice study looking at exomes from children that have autism, showing that if their father was over 40 years of age, they had an increased amount of de novo mutation. But that was only from 0.5 to 1.0 de novo mutations per exome. So, we're seeing a lot of variation that wouldn't meet the amount of variation that we're seeing. I don't have any other genetic suggestion that the mothers are particularly responsible for the malignancy. I think the way that Julian, Logan, and Amy set up the original epidemiology study was to really query perinatal and pregnancy-related risk factors. We do have some trios that we're getting from our hospital, our institution. It's 4 or 5 because it's just such a rare disease. So I don't have enough data there to really say anything that would add to that.
If you go back, first of all, I enjoyed that talk. If you go back to one of the first slides that you showed, you showed a difference between pediatric and adult cancers.
Where'd I go?
Well, if you can't find, my question is,
There. Is that what you're talking about?
well, I don't think so. I think it was before that, but don't. There we go. Isn't that it?
This is just showing an increase in the incidence of pediatric cancers overall over the last 40 years.
Maybe that's not it. But there was one where it was a graph that looked like that, and you talked about, to my left, pediatrics, and to the right was more adult.
So these are the adult cases.
And then there was this group in between, and or was there a group in between? Can you tell us more about the gray zone?
Well, I think, so again, the caveat here is that despite all this genetic mutation that these adult cancers are undergoing, it's because they're genetically unstable. So they're replicating faster, they're just acquiring more hits. So they have a lot more of what we call bystander mutations. So it could be completely true that if there's only 2 or 3 causative mutations per cancer across that entire graph, and so you don't have as much genetic instability in pediatric cancers. They just don't seem to acquire somatic mutation as much as adult mutation. It's not at all to suggest that all of that mutation is causative for the phenotype, because it's not. Well, I think, so if you just ask in principle, why do 90% of cancers happen in people over 60 years of age? And it's because they've had decades to accumulate multiple somatic mutations within the same cell. And so mathematically, that model just doesn't work for a 2-year-old. They just haven't had enough cell divisions to acquire that much damaging genetic change. So it may be true that they need the same 6 damaging genetic changes, but they've inherited 4 of them or 5 of them. So that instead of needing 60 years, they need 6 months to just acquire 1 additional hit, tip the scale over. That's kind of the model that we're trying to work under, and just look for these predisposing profiles. Mm-hmm. Yeah.
There are some adults who, when faced with the option of genetic testing, would forego it because it would lead to a lot of distress.
Yes.
Have you dealt with that issue in your clinic with children who aren't able to make that choice?
Yeah, so most of the children we see don't have cancer and won't get cancer, right? And so it's not at all uncommon for a parent to say, we just don't feel comfortable with this amount of sequencing. And it's for different reasons. A lot of it is due to just concerns about the data being put out there in the public domain, even though we put assurances in the consent form. So there's a lot of reasons why they don't feel comfortable with that, but it happens a fair percentage of the time where they won't. They're usually pretty okay with their child giving a sample and having the child sequenced, but they're actually not comfortable with themselves. I'll shout it out, Logan. You're loud.
For the people
Okay, all right.
So, especially for the AML, the number of hits you had in the MLL genes was so disproportionate. Would you consider that combination deterministic for infant leukemia? And is it ready to move to, clinical use?
Well, so again, the model for cancer treatment has been, let's design a new drug that targets whatever genetic changes are unique to the cancer. But we're proposing that these functional genetic changes are going to be in every cell of the body. So for MLL rearrangements, there's a drug called a DOT1L inhibitor that people are very excited about. It's in preclinical trials. And so, I don't know what that inhibitor would do for somebody that has constitutional genetic hits in some of these genes. It might not have an effect. I don't know. I think what we're seeing, though, in pediatric cancer therapy, there's a growing interest in developing cellular-based therapies to treat these cancers, whether we're converting T cells to come in and attack the cancer cells specifically, or if you use the model of APL, maybe we could get these cells to go to terminal differentiation if we understood the blockages in their development. In single gene disorders like sickle cell disease and thalassemia, there's a growing ability to, in animal models, you can mitigate the disease by taking umbilical cord blood, doing CRISPR swapping of so you can take out the damaged beta globin gene, use CRISPRs to put in a wild type cassette, grow up those cells in culture, and give the animal an autologous bone marrow transplant, and the disease is gone. So, could we do that for infant leukemia, where we could get sequencing from amniocentesis or chorionic villus sampling? You would give the couple sort of a risk stratification of whatever diseases you felt comfortable to model. And, you would say, based on this, I would recommend that you preserve your baby's umbilical cord. And if they do end up with leukemia, we'll have a stem cell population that we can manipulate. So I think there's other therapeutic options that are in the works.
Very good. Let's thank Dr. Druley again.